Another challenge, besides transport, that's faced by all multicellular organisms is communication within the organism. Um, if you're going to coordinate what different parts of an organism are doing, you have to have some way of signaling. In general, in animals, we use electrical signals, but we also use chemical signals. While plants do use electrical signals a little tiny bit, they are much more dependent on chemical signals, or what we call hormones. Hormones are signaling molecules that move around within a multicellular organism, and by doing so end up coordinating the different parts of the organism. And these are some examples of what certain plant hormones look like at the molecular level. These are slower than electrical impulses, but they can be very powerful. Hormones can cause changes in an organism at extremely small concentrations because only a handful of molecules being received or becoming attached to receptors on a cell can lead to a cascade of cellular events that cause large changes in its behavior. There are a couple of different plant responses to the environment that you should know about that are driven by plant hormones. Charles and Francis Darwin actually did an experiment that was much like this with growing grass shoots. These growing grass shoots, these very young shoots, are called coleoptiles. And it's been known for a very long time that when they first pop out of the grass seed underground, they bend toward light. At least they bend toward light, Charles and Francis found out, if you don't cut the tip off or cover the tip with something. Okay, if you cover the lower portion of the coleoptile, it's fine. It just keeps on bending. So what Charles and Francis reasoned, there must be something going on up here at the tip of the coleoptile. Somehow, the tip of this grass plant is sensing light and responding in a way that bends the whole shoot. They also found that if you pass light through certain filters, filters that remove blue light, the coleoptile doesn't bend at all. That's because it turns out, we found in much more recently, it's because the light sensing machinery in a plant is sensitive to blue light. Blue light are the wavelengths that carry the most energy that is used in plant photosynthesis. The way that this seems to work, basically, is that there is a sensing tissue at the tip of a growing plant shoot. When it senses light, it sends some kind of hormonal signal to one side of the coleoptile, which then, so you get a response that is different on the side that's near the light versus the side that's away from the light. You may have done some of these experiments in, say, high school or in other science classes, but a nifty thing that you can do is you can take the tip off a growing shoot and you can put different materials in between that tip and the rest of the plant. If you put something that is permeable to water flow in between the tip and the rest of the shoot will behave perfectly normally, which suggests that it is something that is moving through fluid that causes this change. If you take the piece of agar out of this plant tip after you've done this experiment, and you touch it to the severed side of a different plant shoot, that plant shoot will bend away from that piece of agar. Scientists who originally did these experiments figured out that it's because some water-soluble compound is getting into the agar, and when you take that water-soluble compound and apply it to one side of a plant, it causes the cells on that side to elongate. What's happening is that in the root tip, a hormone called auxin is being generated. It moves away from light and then travels down into the cells that are lower on the plant shoot. That triggers those cells to elongate. And it's that response by those cells, that elongation response, that bends the plant shoot toward the light. Interestingly, auxin functions a very different way in roots. What happens in roots is that if the root is pointing straight down, auxin flows down the center of the root and up all of the sides equally. It's like this upside down fountain going down in the middle and up on the sides. Curiously, if you move a root into a horizontal position, the movement of auxin shifts. Gravity sensing cells change the flow of auxin so that the side towards gravity gets more auxin. And what's really weird about this is that here in the roots, the response to auxin is the opposite. Instead of getting longer when they encounter auxin, like they do in the shoots, root cells actually get shorter. And that bends the plant, that bends the growing root downward into the ground. So you've got these very simple mechanisms. Light-sensitive pigments up at the tips of shoots, gravity-sensitive mechanisms down at the tips of roots that tend to bend plant structures in the directions that they need to go. In most organisms, the coordination of the different parts is not nearly as centralized as it is in human beings. In fact, I would say that for basically all organisms, the way that they function is much less centralized than they are in our bodies. And this is something that takes some getting used to, because we think it was basically the brain is running practically everything. But in most organisms, the different pieces of the organism are mostly left to run themselves. This is true for a lot of the mechanisms that function in plant development, and it's definitely true in the regulation of the stomata. Remember that stomata are these structures around the guard cells, these sort of bean-shaped cells, that determine whether the stomata is open or closed. If it's open, photosynthesis can take place because there's gas exchange, but if it's closed, water can't escape. And those are the two things that stomata need to balance. One of the clever things about stomata is that water loss by the guard cells here tends to cause them to close. When these cells are fully inflated with water, they bend outward, opening up a gap between them. As they become flabby, as they lose water, they tend to collapse in on themselves, shutting the stomata automatically. However, there are other mechanisms. When blue light shines on a leaf, it triggers proton pumps that move ions around in a way that pulls more water into those guard cells. And that causes the guard cells to inflate and open the stomata. So as long as a plant has enough water to that it's not endangering itself basically due to water loss, the presence of light will cause the stomata to open up so that photosynthesis can move forward. What happens essentially is that when blue light strikes a photoreceptor inside those guard cells, a proton pump pushes protons, interestingly, out of the cell. This allows positive potassium ions and negative chloride ions to enter the cell. What happens is the movement of protons actually sets up a proton gradient. And as the protons move back within the cell, that actually pulls multiple different ions in with them. As those ions are pulled in, that lowers the solute potential within the cell. Water is pulled in, and that opens stomata. Senescence is the process of aging, decline, and eventual death of any part of a plant. And plant parts, unlike animal parts for the most part, can die independently of the rest of the organism. One of the chemical compounds that's involved in plant senescence is a compound called ethylene. Ethylene causes fruit to ripen, it causes flowers to fade, and it causes leaves to abscise, or basically senesce and fall off of a plant. That's what happens when deciduous trees drop their leaves in the fall. You may have heard the saying that one bad apple spoils the barrel. And that saying comes from the fact that an apple, as it begins to rot, will start generating lots of ethylene. If you have a lot of apples stored with that one bad apple in a closed space, the concentration of ethylene will build up in that space, and that will trigger senescence on the part of all the other apples. So one of them going bad starts causing all of them to go bad. What happens in leaves depends on, or other plant parts for that matter, depends on the ratio of auxin to ethylene. So auxin is one of these plant hormones that tends to get used again and again and again. At the base of each leaf, there's an area called the abscission zone. And as long as auxin levels are high in the leaf, nothing happens. However, in response to different stimuli, and those can be just the passage of time, they can be day length or temperature or water loss, the auxin levels will go down so that the abscission zone is receiving less auxin. What happens then is that the cells in the abscission zone become more sensitive to ethylene molecules. And that becomes a runaway process that leads that part of the plant to senesce and eventually die. At abscission zones, there's a protective seal that's formed before the leaf falls off. So the plant is not exposing its tissues to things like germs when it drops its leaves. Usually, plant parts, before they drop off like this, also move their nutrients and any sort of valuable chemical compounds into the rest of the plant. So when leaves fall, mostly what they have in them is stuff that is not valuable to the plant, or more often, stuff that cannot be translocated, chemical compounds that are fixed in place. By moving things out, by moving valuable chemicals out of a leaf before it's dropped, plants salvage them. They save most of the valuable material to make new leaves again in the spring.